Welcome back to Biotechnology. Today we're interested in how to get from protein back to the DNA. So what we know is that we can make, or that biology converts DNA into messenger RNA and uses the messenger RNA to make protein. We might want to do that ourselves. We might want to be able to get a new organism to make a protein that we want. That's the whole point of biotechnology. And so we will look at the example that we're going to do. That will be green fluorescent protein, and we would like to get from the green fluorescent protein back to the DNA, because then we will be able to use that to do something. Because otherwise we would have to search through the whole genome of wherever we got the green fluorescent protein from, which is a bacterium that lives in jellyfish arms. But we would like to take a shortcut, and we would like to do that in a biology way, which means we would like to use as many possible biological systems as we can to make it as easy as possible, because otherwise it's going to take a very long time. You might be wondering why we are considering bacteria in this case. But bacteria are special. Bacteria are simpler to understand. We can see the information of the protein in the RNA, whereas in higher species, traditionally known as higher species, the information for the proteins is cut up and mixed in with non-coding DNA, so that it's more difficult. Well, so that it turns out to be more difficult to decode. The RNA codes for it, but the DNA has slices of other information inside. So for a bacterium, we might be able to decode it, but for a higher animal, it's much harder, because we would have to work out what the non-coding sections are and ignore those. Let's get over to the board. See how well this works. Takes longer than I thought. So this would be our green fluorescent protein, and if we go back a step, we would imagine that in a part of the cell that's making green fluorescent protein, there will be plenty of messenger RNA that is coding for green fluorescent protein. And the green fluorescent protein information will be between the start and the end codons for green fluorescent protein. There may be other things on this messenger RNA, because bacteria, unlike other organisms, are not restricted to how many things that one section of RNA codes for. So in our example of the lac operon, one section of messenger RNA codes for not only the enzyme, but also for some other molecules that aid the lactose to get inside the cell in the first place. So they make a pore in the wall and they chaperone the molecule into the cell so that it can then be broken down by the lactase. And all of those things only make sense if you've got them all, so they're all on the same piece of messenger RNA, so that they're all made at the same time, or none of them are made at all. Those things will be coming, the messenger RNA will be coming from DNA. So it will be made from a section of DNA, which in a bacterium, exactly the same as in any other cell, is copied then into messenger RNA. It's the main source of the information, and therefore we will expect, if we want to do something, we're going to have to change the DNA, and we're going to then make messenger RNA from it, which will then make green fluorescent protein. That is our current goal. But where do we get our information from? If we collect the protein that we want, we can sequence it. So we know what the amino acid sequence is in our green fluorescent protein. And then we can predict millions of different potential sequences for our messenger RNA, which we could then try and find, or we could then try and find DNA that is related to it. Or we could find out conditions in which the cell makes green fluorescent protein, and then use that to find out which RNA is turned on at that time and collect that. and then use that to make DNA. But we now have a problem that the typical way that cells work is the DNA is copied into RNA and the RNA is copied into proteins. So we need some other alternative way of doing it, which is a workaround. It's something that is not normally used by cells. It's often used by viruses, however. So if we take this top red arrow, transcriptase is the enzyme that transcribes, so copies, it's effectively there for translating. It converts DNA into a copy of RNA, and reverse transcription is an enzyme that does exactly the opposite job, but it's not really found in organisms, because making DNA from RNA is a really unhelpful thing to do. We want to copy the DNA, and we want to keep its copy quality relatively high so that the daughter cells, in this case it's a bacterium, so it's making daughter cells that are the same as it, the daughter cells will have mostly the same information. Because if it's not mostly the same, then they definitely will die. So we need it to be a good copy. And DNA is better at copying because, as we had it before, we can copy one strand and then check that it's a good copy before it leaves or before it becomes methylated, and then we don't know which strand is which. So if we keep the information of which is the old strand and which is the new strand, then we can get a relatively good copy, and errors are rare. If we make RNA out of it, errors are not going to be as rare because it's a lot harder to check. Once it's gone, once it's let go, it happens easier because RNA doesn't bind very as strongly to DNA as DNA does to itself, then we end up with a problem we can't check to see what it is anymore. So I should put this before. What is a virus anyway? A virus is a piece of information inside a coat of protein. The information for making the protein is on this chain, and also for bundling it up and making the box that it comes in. The protein on the surface has a lock and key mechanism, so it has some way of getting into foreign cells, typically targeted, but not always. So it finds some cell, and by chance, because it doesn't have any movement ability, it doesn't do anything, it happens upon a cell, enters the cell, injects its information, which causes the cell to make more viruses. It causes the cell to copy its genetic material, and it causes the cell to start making its protein and then assembling new virus particles. And in the simplest case, it just carries on assembling them until it bursts, and then they get escaped into the body. Obviously, we can imagine that they get more and more complicated because they then evolve ways of getting into the nasal passages, for example, so that they can escape the body with a sneeze, or causing you to bleed like Ebola does, which also is a way of escaping the body and potentially getting into something else. The very clever ones do even more. So the first type of virus would contain its information as DNA, and then it would need some other way of getting the information off the DNA. It also needs to get the DNA into a cell, so it needs some kind of mechanism for doing that. The cell is likely to fight back, so a higher animal, such as us, has a cell organization like this. We've got a blob of cell, and then we've got a nucleus somewhere inside the cell, which is where the DNA is kept. Outside the nucleus, there is no DNA, and so the cell can contain DNAase, so it contains an enzyme whose job is to break up DNA. DNAase. So its job is to break up DNA, and it's only there, or it's mostly there, as a defense against this type of virus, because if the virus gets into the cell, the DNAase will eat it, will destroy it. That makes it difficult to get the DNA out of the cell, because if we break this membrane, it will start to be attacked immediately. But it is a standard mechanism for avoiding attack by this type of virus. Bacteria don't have a nucleus, and so they are more likely to be attacked by this type of virus. So we can imagine if a DNA virus attacks a normal cell, or not a bacterium, it has a job of getting its DNA into the nucleus, so it has to do something about that. If it was an RNA virus, it might not have to do that. And if it's a DNA virus, it still has to get its DNA somehow copied into RNA, so it needs to bring some transcriptase along with it, or trick the transcriptase of the cell into doing the job for it. So depending on whether it is going to insert its DNA, then it can use the cell mechanism of the host cell, or if it's not going to do that, it needs to carry along or make its own, but probably it needs to carry along some enzymes of its own to transcribe the DNA into RNA, which we might be able to use later for some kind of other purposes because it's specific transcriptase for virus short sections. Okay, back to this and to correct a few things that I said wrong. So the DNA viruses, they use mostly the cellular machinery inside the cell, but RNA viruses can't do that because they don't have DNA. They have RNA in them, and typically they will have either sense RNA or antisense RNA, or they will be double-stranded. So the first thing that they have to do is make new RNA, copy themselves. And so they need an enzyme. Let's put a random cell in there. They need an enzyme that can copy, oops, that's backwards, but it doesn't matter. Let's have it in both. They need an enzyme that can copy the RNA, and it can copy it by making a double-stranded RNA, which means it has to make an antisense strand from the sense strand and a sense strand from the antisense strand. The antisense strand cannot make any protein because it's got the wrong code on it, and the sense strand obviously can. If we have a sense virus, then its RNA can immediately make protein. Let's put that blue. So this can immediately make protein. So it gets off to a running start. The antisense virus can't do that. It has to first of all make a sense strand, which then makes protein. But it has a different advantage that when it comes to making new virus particles, when it's got the protein and both sets of RNA, then it can pack away the antisense strands, and the sense strands can carry on making protein. So each of the two parts of the RNA have a job. Whereas for the sense RNA, the antisense strands do nothing. They don't make protein, and they don't make new virus particles, so they're waste. They could just get in the way. So that is a bit of a problem, or it's a bit of a problem. It means that the sense viruses get off to a faster start, but they then don't have such an efficient reproduction, and the antisense viruses are the other way around. They get off to a slower start, but they work better afterwards. But we do have a new molecule that the viruses have to have invented, they have to have generated, and that is something to make double-stranded RNA, to copy RNA, which is very useful if we want to copy RNA. So now we have a molecular mechanism to copy RNA that we can steal from viruses, should we want to do that. But the type of viruses most interesting to biotechnology are the ones that insert their DNA into the host DNA. So they insert some information into the host DNA and then they come back later. Things like the HIV, I nearly said HIV virus, which would be doubled, but things like HIV have a part of their genome that they insert into the human genome, so they insert it into the human DNA, and then they can be resurrected effectively even if they're completely wiped out from the body, that cell can start suddenly making virus particles again because it's stuck in the DNA. So that's a useful property that viruses might have. It's not useful to us yet, but we might want to hijack it and see how it works. The most interesting and complex version starts off with a piece of RNA, and it converts the RNA back into DNA. So if we take our RNA and we have a new molecule, which is our reverse transcriptase, that's our T, then we can convert that back into DNA, which I'll make red because I was making the second strand red before. But there are two interesting factors about this. The first interesting factor is that the reverse transcriptase destroys the RNA as it goes along. There's no logical reason why it has to do that, but because it doesn't matter for biology to work, it's not evolved out. There's no reason for it to stop happening, and so it does. So effectively, it starts to copy the opposite strand, and then it deletes the RNA strand. So it starts to make the opposite strand, and then it starts to delete the RNA strand to make two double-stranded DNA. I'm not entirely sure whether it goes all the way along and comes back. I think it does it from the same end. This makes our DNA, and this is very useful for us because then we can do things. So if, for example, we want to find the gene for green fluorescent protein, we know it's a short gene, we can find some RNA for green fluorescent protein, and we can make DNA out of it using reverse transcriptase. There is a problem, the other thing that I forgot to mention, which is reverse transcriptase isn't very good at copying. Why isn't it very good at copying? Well, if we copy this strand and we make an error, we have no way of going back. We've destroyed the RNA. And although we know which is the old strand and which is the new strand, there's no other molecule, there's no molecular mechanism to repair that damage if it happens. This is potentially useful to the virus because it can then mutate more quickly, which might make it more difficult for the host organism to get round to evolve a defense to, but it also might cause it to not work some of the time, much of the time. So it's a double-edged sword, effectively. This error proneness is also present in the double-stranded RNA viruses and the types of RNA virus, the single-stranded RNA viruses that go via a double-stranded copy, so they don't use a DNA mechanism, because they also use a poor copying mechanism. They have a not as good copying mechanism, and so they mutate more rapidly than other viruses do. That makes them in some ways more robust, but in some ways prone to failing. It isn't the only use of this reverse transcriptase, however, because biologists are very good at exactly this, pressing things into doing new jobs, pressing biomolecules into doing things that are useful to us rather than harmful. And so in this case, we might consider the current pandemic. If we are infected by a coronavirus, so this would be one type of coronavirus, might be COVID-19, might be a different coronavirus, there is no DNA. It's only RNA, it's an RNA virus, so we've got only RNA that we're looking for. But if we find the RNA and we reverse transcribe it, so we need reverse transcriptase, then we get DNA for the virus, which doesn't normally exist. But the good thing about DNA is that we know to PCR. We know what to do with it. We can do polymerase chain reaction that has also been modified by humans to work better. So we need a primer, a forward and a reverse primer, that are linked to the sequence of the RNA that we're looking for. And we don't need to search for the whole genome of coronaviruses. We just need a section. Then we grow it, and we can grow as much as we like. What we can also do is we can put several other primers for different primers for different coronaviruses, and so we can measure for COVID-19 and also for flu at the same time and tell the difference, because it's the primers that determine whether we copy it or not. There was a bit of controversy in the high copy cycle, but that is running this over many thermal cycles, which means that the accuracy of how much there was at the beginning goes down massively, because obviously we multiply by 2 and multiply by 2 lots of times, and every doubling is not perfect, so it's probably slightly less than 2, and therefore we don't know how much RNA we had at the start if we copy it too often. But it doesn't mean that it's useless, because we're looking for, is it there or not? It depends what the test is for, whether it matters what the accuracy of the number is. In this case, we're looking for a very sensitive test, but we are not worried quite so much about how accurate it is, because does it matter whether there were 10 copies of RNA or 100 copies of RNA in the original sample? Not really, because of the way that the sample is taken. It's not very reproducible. You might hit some cells that have a lot of the RNA in, and then you get a big sample. You might hit one, in which case you'll be lucky to get it to reproduce. And so this particular type of test is more variable at the sample harvesting part, and so therefore we can copy it up a lot of times and say, well, we don't know anyway. This is similar to the difference between a pregnancy test and a drugs test. In a pregnancy test, the molecule that we're looking for, human gonadotrophin, is present in female human urine anyway, and so we need to measure the amount. We need to measure a cutoff amount. Below this, it should measure negative. Above it, it should measure positive. We can't do a test like this where we're just looking for, is there any at all in there, because otherwise every test would come out positive. So we've got a difference. We've got a different style of test, and we need to be more careful about the amount. In that case, we don't have to be super accurate, but we still need to be more careful about the amount, because if you put in twice as much P, then we have a different result. And so the test is set up in order to take a certain amount of sample. It doesn't matter how much is in the beaker of piss that you put this test into, it helps itself to a measured amount. It's how the test works. We'll look into that later when we look at antibodies. Obviously, while we're here on viruses, and we've talked about HIV that inserts its genetic material into the human genome, then we can see that those would also be useful as a molecular tool in order to cause biotechnological changes in things. So we could take a virus, we could change its information. Instead of coding for the protein in the coat, it codes for something that we want, and we can make another cell, a new type of cell, start to make that protein. The mechanism for that is much more complicated and much more difficult to do, so the insertion mechanism into the genome is easier to steal as a whole thing. So we typically use a virus type, but then we end up with limitations because viruses tend to infect certain tissue types. So if we need to affect lung tissue, then we need a type of a cold virus that will infect lung tissue. If we need to infect other types of tissue, we need to find a type of virus that affects that type of tissue, because it's otherwise difficult to modify it to get it to do that, and it might tend to still target the tissue that it would normally do. So the topic for today was transcription and reverse transcription, and I thought I would do it this way around for a change. So we've got our natural system where viruses either copy their RNA into double-stranded RNA and then back again, so they split it again into single-stranded RNA, or they reverse transcribe their RNA into DNA, or they just use DNA, and hijack the cell. I said it wrong at the beginning, but they actually hijack the cell's DNA copying and conversion into RNA machinery, which is obviously more efficient for the virus. It can be smaller, but DNA is bigger than RNA, so it's a trade-off. And the DNA has to get into the nucleus if it's an animal cell. So we've got those effects, and we can use the reverse transcriptase in order to convert RNA into DNA if we want to copy it up for a test, or if we just want to have DNA for a protein, because it's a lot easier to find the RNA. It's already cut into the correct sections, it's already ready, and we know where the stop and the start codons are because they are specific sequences, so we can find those and we can make a DNA sequence out of it using reverse transcriptase, which is hijacking the hijacker's system against itself. We're using the virus's evil mechanism, its survival mechanism, for other purposes. Okay, I think that's all we've got time for today. Next time we'll do a different topic again.